Hello, friends. This is Dr. Nair Kazmi again with another episode of COVID-19 infections. And in this particular episode, we're going to talk about how our immune system reacts to this virus and whether reinfection is possible or not. So, to start with, we need to see how our immune system is made. And I will start with drawing one of the human cells. And in this particular case, I would say this is one of the cells. And let's say this cell is one of the type 2 pneumocytes in the lungs alveoli. And it has got typical receptors on its surface, which are known as the ACE2 receptors, which are responsible for binding this virus to the cell surface. Now, this is a coronavirus, and these are the surface spike proteins on the surface. It has got a single stranded RNA inside it. So what happens is when this virus comes and attaches to the surface proteins, after that the virus is engulfed inside, and there what happens is that the viral RNA comes out of the cell, and then it gives the cell ribosomes a message to make copies of this RNA. So many copies are formed, and then these copies, from these copies, further virus particles are made, and then they are released from the cell. So from an infected cell, you get many copies of the viruses that come out, and then they infect the other cells. Now each of this virus has got this RNA inside, and this RNA comes from the multiplication of the original RNA when the original cell was infected. Now, typically, in an acute COVID-19 infection, so far, the basis of diagnosis is based on the detection of this RNA in the human blood. And the method used to detect this RNA in the blood is known as reverse transcriptase PCR, and this stands for the process in which this enzyme, which is known as reverse transcriptase, what it does is that it does a reverse transcription, that copies of DNA are formed from the RNA. So if this is the viral RNA, with the help of this enzyme, what happens is that many copies of the RNA are formed by amplification, and as this DNA contains a particular code, that code is detected by one of the probes, and if it reads and if it matches that, then it gets highlighted, it gets flagged, and bam, there you get a positive test. So your PCR is positive, and you know that this person is infected. But let's say if it is not able to match the code, then it would not flag up, and you get a negative test. So the basis of the testing is on this real time reverse transcriptase PCR. Now the problem with this is that recent methods, epidemiological methods, have shown that the sensitivity of this method is somewhere between 70 to 80%. And what does that mean is that in around, if let's say if 100% are actually infected with the COVID-19 virus, and you test all of them with this RT-PCR method, so it would come out to be positive in 70 to 80% of the cases. So you still would miss around 20 to 30% of the cases which would be infected, but their PCR would be negative. What that means that they are false negative. Okay, and then these might be the people who might be spreading it to other people. So that's why it is imperative that some other form of testing is also developed, okay, which actually shows us whether the immune system is working against the virus or not. Now, normally what happens is as the disease progresses, the human body starts making antibodies. So for example, if this was the original infected cell and the virus came and infected this cell, so as the surface protein of the virus attaches with the surface proteins of the cell, some of them virus particle, they would remain, the virus spike proteins, they would remain on the cell, and this would flag up the cell. So what would happen is that the some of the arm of the immune system, they will recognize these surface proteins, okay. And it's a complex method in which what would happen is that the helper T cells, okay, they would come, they would recognize these cells, okay, and they would present these antigens to another component of the immune system, that are the B lymphocytes, and those B lymphocytes would then produce specific antibodies against these surface proteins. So antibodies are shaped like this, and this is the constant portion of the antibodies, and these are the variable ones, and then this variable one that usually attaches to the surface proteins like this, and then in turns would activate the other arm of the immune system, like the complement system, the neutrophils. They would rush into this area and they would destroy the infected cells. So over a period of time, more and more of these antibodies would develop. So normally in the acute phase of the disease, the antibodies which are made are of IgM class, and as the disease finally settles down, we have got memory cells, okay, which make another class of antibodies which are known as immunoglobulin G, which give you long lasting immunity against this virus. So in case if this virus comes again in the body, these antibodies would be already there, they would immediately coat those cells, okay, and the immune system would be activated and they would immediately destroy the cell before the virus replicates and infects more cells. So then the disease is arrested in its initial state and the person does not develop a full-blown disease. So that is very important. Now testing for these antibodies is also important, because if the testing shows that you are positive for either the IgM or IgG, it means you have developed immunity against the particular virus. If I draw a graph, so in the early stages of the infection, so here is where you get the PCR positive. So the PCR is positive and at some point in time, okay, as the infection progresses, the immune system starts kicking in, and then you start developing antibodies, and the antibody starts becoming positive in the blood. As I said earlier, that initial phase would consist of IgM antibodies, and the late phase of the disease would contain the IgG antibodies. Usually it happens after the recovery, and the presence of these antibodies can actually tell you whether the infection has been an old one or whether there has been a reinfection. And so recently they did a study in which they tried to see if a reinfection is possible in case of COVID-19 or not. And this study was done in Chinese rhesus monkeys, and the rhesus monkeys are closest to humans in terms of their genetics. So 4 rhesus monkeys were taken, and they were infected with COVID-19, and this research was recently published in Biorexiv journal. And to simplify it, what I would do is I would make a diagram to explain that. So what they did was that they took 4 monkeys, and all of them, they were infected with COVID-19 virus, and then they followed them over a timeline. So what they found out in the initial phase of the infection was that all of them, they had a mild temperature rise, which was maximum on day 3. They had some weight loss, and they had other mild symptoms of the disease. Now what they did was, on day 5 they actually sacrificed one of the monkey, and then they looked at different tissues to see if viral replication was there. So they took out different organs and they looked for virus replication in that, and they found out that the viral replication was present in the lungs, obviously, which was the major area of replication. Lungs, heart, kidneys, spinal cord, gut, and bladder. So the virus was replicating in almost all of the tissues of the body. What they did was then they followed the rest of the monkeys on day 7, day 14, day 21, and day 28. So they found out here all of them were PCR positive. All of them were positive. But then on day 14, the PCR was negative. They start become negative in all of these 3 surviving monkeys, and they also had start developing antibodies. So the antibody was positive here, was positive here, was positive here. So it kept on. So throughout on day 21, day 28, they found out PCR was negative in all these 3 cases, and antibodies were positive. On day 28, what they did was they reinfected these 3 surviving monkeys again with the COVID-19 virus, and again they tried to find out if they were PCR positive or not. So they had very mild elevation of temperature, but the PCR, when it was done, was negative in all the 3 monkeys. So what they did, then they again sacrificed one of the monkey, and they looked at the same organs to see if there was any viral replication, and they found out that there was no evidence of viral replication in lungs, heart, kidney, spinal cord, gut, bladder, or skeletal tissue. And they also find out that in the rest of the 2 monkeys, the immunoglobulins titers had risen. So the immunoglobulins had increased. So they concluded from this research that reinfection because of COVID-19 was not possible in rhesus monkeys, and they have generalized that because they're quite close to the human beings in terms of genetic. So most probably it's not possible for COVID-19 to reinfect the individuals, and probably the immunity coming in from the infections would be long lasting. And this is one of the reasons that immunoglobulin based testing actually helps in further stratification of the cases. And recently today I was told that soon here in UK also the antibody based testing would be provided. I've heard that they would be provided in boots as well, and it's simply based on finger prick testing, and people can even test it at home. I don't know how much truth is this in this news, but personally I think that there is a potential for the antibody based testing, because it's cheaper, it's easier to do, and it can complement the diagnosis with the PCR, and can give us long lasting data in terms of how the immune system responds to this viral infection, and whether any reinfection would be possible in the coming days or not. So thank you very much. If there are any questions, comments, or queries, please feel free to ask me. Do comment in the space below, and if there is any issue, you can ask me for clarification. Thank you very much.